Hi everyone. In this video, we're going to start chapter 20, which is all about enzymes. So enzymes are proteins that catalyze chemical reactions. So they speed up chemical reactions by lowering the activation energy for the reaction. So down below, we have a kind of simple graph here. We've got reaction progress on the x-axis and energy on the y-axis. So in a reaction, you have your reactants at one energy, and then when they start to react, they need a certain amount of energy in order to react with each other. So that's going to give us that activation barrier, and they have to get over this barrier in order to form the products. But an enzyme is actually going to help the reaction occur sometimes more quickly or more often. So if you have an enzyme available, it acts as a catalyst, and even if your reactants start at the same energy level, it's going to lower that energy barrier needed to get to the products. So this is kind of similar to, let's say you're asked to clean up your room. And if you do it by yourself, that's going to take a lot of energy and time. But if you ask a friend to help you, they can act as kind of a catalyst or enzyme. They can motivate you, they can help you clean, and it will go much more quickly. So kind of the same thing here with enzymes. They're sort of like friends that are helping out a reaction. So enzymes are specific in the type of reactions that they catalyze. So there's a few different what we call specificities here. So these are the types of reactions they catalyze. So there's absolute specificity, and that's where an enzyme acts only on one substance, and that substance will be called a substrate. And that's true for all of these different interactions between an enzyme and a substance. So a substrate is the substance that undergoes a chemical change that's catalyzed by an enzyme. So we'll sometimes refer to these substances as substrates. Now, relative specificity acts on structurally related substances. So this type of enzyme wouldn't be as picky. It might act on multiple sugars, for example. And we saw that in lab. We saw that yeast enzymes can act on many different sugars, just as long as they're somewhat related, right? But in absolute specificity, an enzyme would only act on, for instance, glucose and not any other sugar. Then there's stereochemical specificity, and this is an enzyme that distinguishes between stereoisomers. So let's say you had a D sugar and an L sugar. An enzyme that had stereochemical specificity might only act on the D sugar and not the L sugar. So down below we have an example of an enzyme and its reaction with a substrate. Our substrate is urea, so this is present in urine, and the enzyme urease will break apart urea to form carbon dioxide and 2 ammonia molecules. Now, if we just had urea react with water and no enzyme, this process would probably take a really long time. But with the presence of the enzyme urease, I think I'm pronouncing that right. It sounds weird, but it makes the reaction quicker and it occurs more easily. So there's different ways that we can name these enzymes. So there's an old naming scheme, and this was when we didn't know about a lot of different enzymes. We only had knowledge of a few enzymes. So the earliest enzymes have names that end in IN, and that just indicates their protein composition. So we've got, for instance, pepsin, trypsin, chymotrypsin, and notice they're all ending in IN. But once we discovered more and more enzymes, we needed a better way to name them, or a more systematic way to name them. So the new systematic nomenclature system is the Enzyme Commission System, or EC. So this has 6 major classes that are based on the type of reaction that's catalyzed, and we'll look at that chart in a second. But basically, in this naming scheme, we're going to name the specific substrate or substance that the enzyme reacts with, and the functional group that's acted upon, as well as the type of reaction that's catalyzed. So there's a lot that goes into this naming scheme. And then we end the name in ACE. So let's look at the 6 major classes. So in your textbook, there is this table for the classification of enzymes. So the first class is oxidoreductases. So these are enzymes that help with oxidation reduction reactions, and they just help those reactions occur more quickly. The second class, transferases, these help to transfer functional groups from one part of a molecule to another, for example. Then we have hydrolases, and these help with hydrolysis reactions. Then we have lyases, which are enzymes that will help to add something across a double bond or remove something from a bond to create a double bond. So we saw some of these reactions really early on in this class. We saw that we could add, for instance, H and OH across a double bond. We could add 2 hydrogens across a double bond. So these are enzymes that help with that process or the reverse of that process. Next is the class isomerases. So these enzymes help with converting an isomer to a different type of isomer. So for instance, remember way back we talked about cis and trans isomers. So the conversion of a cis isomer to a trans isomer would typically be kind of slow. You need a lot of energy to do that. But if you have an enzyme handy, that can make the process much quicker. So those help with that and other similar reactions. And then finally, we have ligases, and these are enzymes that help with the formation of bonds with ATP cleavage, which probably doesn't make a lot of sense right now. We'll talk about ATP later on, so don't worry about that too much. Okay, so let's kind of look at an example of how to name an enzyme using that EC system. So this is that same reaction we saw earlier where we're reacting urea with water, and then we had an enzyme that helped to break apart urea into carbon dioxide and 2 ammonia molecules, NH3. So the EC name is urea amidohydrolase. So again, we're going to first list the substrate that we're reacting with, so that's urea. Then we name the functional group, which was amide, so that's the amido part. And then we also list the type of reaction that we're doing, which in this case is hydrolysis, because we're reacting with water. So we've got urea, then amide, which we changed to amido, and finally hydrolysis, which we then called hydrolase. So remember, we end the enzyme name in ASE. So I'm not going to make you name enzymes, but this is just to point out that you can kind of figure out what an enzyme does just by looking at its name using the EC system. So urea amidohydrolase, we could say, okay, that means we're acting on urea, so we're doing some sort of reaction with urea. Amido tells us, okay, we must be doing a reaction with the amide group in urea, so this group here. And hydrolase would tell us, oh, we must be doing hydrolysis, so we must be reacting with water. and kind of breaking it apart. All right. Now, of course, the EC system is kind of complex, so there's also common names that we might use. And common names for enzymes are shorter than the EC system names. So, for instance, we could just list the substrate with ASE on the end. So if we go back, here we're acting on the molecule urea. So we could just call the enzyme urease. So we just added ASE to the end there. So that's one method that you could use to make it easier to name the enzyme. Or you could add ASE to a combination of the substrate name and the type of reaction. So one example is actually given down below. So the substrate alcohol and a dehydrogenation reaction type would give us a common name of alcohol dehydrogenase. So we named the substrate, the type of reaction, and then we just end it in ASE. So that's also a nice thing with enzyme naming. No matter what method you're really using, typically enzyme names either end in IN or ASE, so you can kind of figure out, oh, this must be an enzyme. All right, so let's actually talk about what enzymes do. Let's talk about their mechanism. So all enzymes have an active site. So this is the location on the enzyme where a substrate will bind, and that's where catalysis or the catalytic process occurs. So for example, we've got our enzyme. So it might look, kind of globular, for instance, something like that. And then we've got our substrate. So the substrate is, let's say it just kind of looks like this. So the enzyme's active site is right here where the substrate could kind of fit in. So eventually you'll form what's called an enzyme substrate complex. So let's see if I can kind of redraw this here. So we've got our enzyme, and then the substrate is just going to fit right into that active site. So now this is our enzyme substrate complex. Then the enzyme does its work. It, does whatever it needs to do to help with the reaction. And then the two are going to split apart. So then we end up with our enzyme again by itself. So it could be reused in another reaction. And then we'll have our product. So maybe the enzyme turns our substrate into a different product here. And that's really it with enzymes. Now, of course, you could zoom in and look more at the specific reaction that's occurring between the enzyme and the substrate, but we're not going to go into the nitty-gritty there. We just kind of want to look at the overview of what enzymes do. So let's look at a specific example. So let's say that we've got an enzyme sucrase. So notice we just took the name sucrose and added ASE to it. And our substrate, of course, is sucrose in this situation. But the enzyme sucrase is going to have an active site that sucrose fits into. So then once sucrose is in the active site, that's when the enzyme can do its work. And remember, sucrose is actually made up of two monosaccharides. So let's just kind of draw a generic sucrose molecule there. So the enzyme is going to eventually connect with sucrose, and then it's going to do its magic and split sucrose apart. So then we'll end up with the products glucose and fructose, and the enzyme will just be left over to go break apart other sucrose molecules. So again, we would end up with glucose, fructose, and then our enzyme. I always kind of draw enzymes as little kidney beans. I don't know why. Okay, so some enzymes do require their own help in the form of something called a cofactor. So this cofactor is required for some enzymes to function properly. It's not a prosthetic group necessarily, like what we've talked about before with like hemoglobin, for instance, and heme. So this cofactor is only weakly bound to the enzyme. It's not stuck in the protein. So cofactors could be a non-protein molecule or an ion. If the cofactor is an organic molecule, we actually call it a coenzyme. So let's say you have just the enzyme without that cofactor. We would call that an apoenzyme because it's missing that cofactor. But if you have your enzyme without the cofactor, so the apoenzyme, and then you add the cofactor in, that's when you get your active enzyme that can actually do its job. So some enzymes do require this little bit of extra help. And here are some examples of enzymes that have a cofactor. So carbonic anhydrase, that requires zinc 2+. Alcohol dehydrogenase, which breaks down alcohol, that also requires zinc 2+. We know previously we saw hemoglobin is a protein that requires iron. And then you can see there are other examples down below as well. So a lot of the time a cofactor is just an ion, a metal ion, but sometimes it might be a more complex molecule that's needed for the enzyme to work. And often those more complex molecules are derived from vitamins, and that's why vitamins are really important for us, because if we want our bodies to function properly, sometimes we need those vitamins so we can, so our enzymes can work. So now there are two main theories on how substrates fit into the active site of an enzyme. The first one is called the lock and key theory. So this is pretty self-explanatory. This theory states that the substrate has a shape that exactly fits the active site, so like a key in a lock. And this would explain why some enzymes specifically react with just one substance, right? So we've got this rigid enzyme, and it can only fit something with this shape. So the substrate has to have that shape in order to fit into the enzyme. Now there is one other theory called the induced fit theory. So this states that the conformation of the active site changes to accommodate an incoming substrate. So this is more earlier we talked about how some enzymes just need like an amide group, or maybe they just need to have a sugar fit in there. It doesn't have to be a specific sugar. So that would be this, for instance. So let's say our enzyme just kind of has a generic little active site there, and the substrate comes in and forces the enzyme to change its active site shape to accommodate it. Now, again, these are theories, and there's a lot of research that's done on enzymes. So, if you look at a specific enzyme, you can kind of see, how does the substrate fit in? Does it accommodate other substrates or just that substrate? So, these are theories. All right, so as review, let's draw pictures for those two enzyme mechanisms that we just saw. So pause the video, and without looking back, see if you can draw a picture of the lock and key theory and the induced fit theory. And then when you're done, you can unpause the video and we'll go over it together. Okay, so let's start with the lock and key theory. So this one's pretty easy, right? We've got our enzyme, and let's say its active site is shaped like a square. And let's say our substrate is conveniently shaped like a square. So then, if they join together, we've got a perfect lock and key. So if we had a different shape, let's say maybe we had like this shape, that wouldn't fit, right? So now let's draw a picture of the induced fit theory. So let's say the active site is shaped like a square again, but maybe this time the enzyme can change its shape to accommodate something different. So maybe we have a triangle. So then if the enzyme changes shape, its active site could become a triangular shape to fit our substrate. And obviously, molecules aren't shaped like squares or triangles, but you get the idea. Okay, so we'll stop there. In the next video, we're going to talk about factors that affect enzyme activity. So I will see you then.